Soil is one of the most important factors for the success of plant growth. Soil nutrients such as nitrogen, phosphorus, and potassium directly affect the plant growth and density. Nitrogen is usually found in all plant cells, including their proteins and hormones. According to Hewitson, when there is an increased level of nitrate in the environment, the osmotic concentration also increases. This allows for plants to take up more nitrate, which eventually results in a higher plant density. Our experiment was based on the diversity of Ball State Forest, and we hypothesized that an increased resource availability increases plant density. Thus, our null hypothesis is that the amount of resources available does not influence plant diversity. Finally, we predicted that if there is a higher nitrate concentration, then more plants will be present.
On November 4, 2017, we collected our first sample. To determine where the plot would be, we used a tape measure and a 1-meter plot. We walked down the path and stopped at a random location diverging from the path. Then, we used a tape measure to measure 2 meters into the forest. With that, our plot was chosen. Moreover, we cleared any debris, counted the number of plants in the area, and took small samples of each plant in the plot for further identification.
We used a metric ruler, a large shovel, a small shovel, and a plastic bag. First, we chose the largest plant in the plot and dug a small hole 15 centimeters in depth. We took a couple scoops of the soil from this depth as our soil sample and placed it into a plastic bag. We continued these methods for our next 5 plots on November 9th, and we walked about 1.6 kilometers into Ball State Forest for our last plot.
We tested with the ChemMet's nitrate colorimetric test kit. We sifted the soil and measured about 8 grams. We added about 12 milliliters of deionized water and stirred. 1.5 milliliters of this mixture was added to the testing tube and diluted with 9.5 milliliters of deionized water. We then followed the remaining procedures as directed by the test kit. And lastly, we compared the color of the ample with the spectrum provided by the kit. This test was repeated for the remaining soil samples.
Plant abundance was measured as the total number of plants in each 1 meter squared plot. It seems that the further away from the road that we traveled, the more plants are present in each plot. This could be because most plants are selected species, so they produce more offspring in order to increase the survival rate of adult plants. This could account for the high number of individual plants in plots 4 and 5.
Next, we compared the relationship between plant density and nitrate concentration. It was discovered that there was a positive relationship between nitrate concentration and plant density. Then, we examined the relationship between species richness and nitrate concentration. Species richness was measured as the number of different species in each plot. Here, there is a slight positive relationship between species richness and nitrate concentration of the soil.
There were 3 invasive species that were found in our plots: garlic mustard, Alliolaria petiolata, the Japanese honeysuckle, Lonicera japonica, and the baby or Japanese rose, Rosa multiflora. Garlic mustard and the Japanese honeysuckle were the most commonly occurring species among our plots. Garlic mustard uses allelopathy, which is when a plant produces and excretes noxious chemicals that inhibits other plants from growing. There are several confounding factors that could have influenced the plant diversity and species richness in Ball State Forest. For example, plants depend on other resources besides nitrate in order to grow. The specific resource conditions of the area in which the plots were made could have also affected the nitrate concentration.
In conclusion, our hypothesis that an increase in resource availability increases plant density was supported. We saw a positive relationship between nitrate concentration and the number of plants. In order to make our data more accurate and precise, we should have taken more samples from each of our plots. Then we could have calculated the average nitrate concentration. Additionally, it would have been easier to understand the variance in nitrate concentration, plant density, and species richness if the distance from the main road to our plots was measured.